Whether you realize it or not, you are a highly sophisticated fortune teller.
That's a pretty bold claim to start us off.
Right. But think about it. When you navigate a busy intersection or anticipate the exact drop of a song.
Or guess when that consistently delayed train is finally going to pull in.
Exactly. Your brain is doing this impossibly complex calculus in milliseconds. You navigate daily life under an immense amount of uncertainty, yet somehow your biological hardware flawlessly predicts the future.
It's completely seamless to us, which is why, we rarely stop to appreciate the sheer computational power required for those everyday predictions.
Yeah, we just take it for granted.
We do. But what your brain is actually doing is taking in ambiguous, noisy information from the environment and cross-referencing it with past experiences to guess what happens next. In neuroscience and math, this is known as Bayesian inference.
Right. Bayesian inference. And today, we are taking a deep dive into an absolutely fascinating April 2026 paper from Nature Neuroscience by Koppen, Narain, and their colleagues.
A really groundbreaking piece of literature.
It really is. The mission for us today is to uncover the exact neural circuits that allow your brain to internalize the statistics of our uncertain world.
Because while we've known for a long time that the brain behaves in this predictive Bayesian way.
Right, the behavior's obvious.
Exactly. But exactly how a wet, squishy mass of cells actually does this math and where it happens structurally, that's been a massive mystery. Until now.
Okay, let's unpack this. Where do we even start looking in the brain for this?
Well, to solve the mystery, the researchers zeroed in on the cerebellum.
That's the part of the back, right?
Yep. If you picture the brain, the cerebellum is that distinct, heavily folded structure nestled at the back of the skull, just beneath the cerebral hemispheres.
Textbooks usually just call it the motor control center.
Right, for coordination and balance.
Yeah.
But over the years, we've realized it is also the brain's premier timekeeper. It's deeply involved in precise timing and sensor-motor predictions.
Okay, so to figure out how the cerebellum learns the probability of future events, the researchers set up this incredibly elegant behavioral experiment.
It's called probabilistic trace eyeblink conditioning.
Right. Trace eyeblink conditioning. I want to make sure I'm visualizing the setup correctly because the exact parameters are crucial here.
Let's walk through it.
So we're looking at mice that are head fixed. First, a light flashes in their field of vision. This is the conditioned stimulus, basically a warning sign.
A visual cue that something's about to happen.
Right. Then there's a temporal gap, a period of total silence. And finally, a gentle puff of air is delivered directly to the mouse's eye.
Which is the unconditioned stimulus.
Exactly. And naturally, that air puff causes a reflexive blink. But over time, the mouse learns the sequence, right?
It does. It starts to blink before the air puff even arrives, squeezing his eye shut in anticipation. We call that the conditioned response or the predictive response.
Okay, but under standard conditioning, that gap of silence between the light and the puff is like a fixed amount of time.
Yes, exactly the same every single trial. The mouse learns to predict it down to the millisecond.
But real life isn't like that. Real life is messy.
Which is exactly why the researchers introduced a fascinating twist: probabilistic timing. They threw out the fixed time gap.
Right. Instead, they drew the timing of the air puff from different prior probability distributions.
So they basically created different statistical environments for different groups of mice.
I like to picture this like a temporal roulette wheel. But instead of numbers on the wheel, you have different time delays.
That's a great analogy.
So they set up 5 different roulette wheels. First, the single distribution.
That one is a 100% chance the air puff hits exactly at 350 milliseconds. Highly predictable.
Then you have the narrow distribution, which is a 33% chance spread across a very tight range around 350 milliseconds.
Right, just a little bit of fuzziness.
But then gets crazy with the wide distribution.
Yeah.
This one is just a 20% chance of an air puff spread broadly across anywhere from 200 to 500 milliseconds.
Highly uncertain.
Exactly. And they also had a short control group, 100% chance at 200 milliseconds, and a bimodal group.
The bimodal one is key later. It had 2 distinct peaks of probability. A high chance at 200 milliseconds, a lull, and then another high chance at 500 milliseconds.
So to see how the mice adapted to these totally different environments, they didn't just check if the mouse blinked.
No, simply blinking isn't enough data. They looked at the highly specific kinematics of the eyelid closure.
The actual physical shape of the blink.
Exactly. They measured 4 crucial metrics. Let me give you a way to visualize this, like a sprinter on a track.
Okay, I love the mnemonics. Let's hear it.
First is T onset, When does the runner leave the block? Or in this case, the exact millisecond the eyelid starts to move.
Got it. T onset is leaving the block.
Second is peak velocity. How fast is their top speed? Meaning, how fast does the eyelid snap shut?
Makes sense.
Third is ACR, or amplitude. How high do they jump? Basically, how tightly is the eye squeezed shut?
Okay, and the fourth one?
Fourth is AUC, the area under the curve. How far did they travel overall? This tells us how intensely the eye stayed closed over the entire predictive window.
Okay, T onset, velocity, amplitude, and area under the curve.
Yeah.
When they looked at those metrics across the different roulette wheels, the findings were stark.
They really were. As the distribution got wider, meaning more uncertain, the physical blinks adjusted perfectly.
In that wide distribution, the mice had much earlier T onsets, higher peak velocities, larger amplitudes, and larger total areas under the curve.
They basically kept their eyes squeezed shut longer and harder.
But hold on, I got a problem with this conclusion.
Oh, what's the issue?
If the danger window is massive like, an air puff could come anywhere from 200 to 500 milliseconds, isn't the mouse just flinching at the 200 millisecond mark because it's terrified?
Ah, the panic theory.
Yeah, like if I know something might hit me soon, I'm just going to brace myself as early as possible. How do we know the brain is actually learning the full, complex mathematical probability curve?
That is a brilliant critical thinking question. And it's exactly the correlation versus causation problem the researchers had to solve.
So how did they prove it wasn't just panicking?
They pointed to that short versus wide control experiment.
Remind me, the short condition was a 100% chance at 200 milliseconds.
Correct. And the wide condition also begins its danger zone at 200 milliseconds. So if the mice were just panicking at the earliest possible threat, the blinks in both groups would look identical.
Oh, because they'd both just flinch at 200.
Exactly, but they didn't look identical at all.
What was the difference?
The mice in the short condition produced very narrow, rapid blinks that reopened quickly. They were perfectly tailored to a single event at 200 milliseconds.
But the wide ones?
The wide condition mice produced sustained, prolonged eye closures that stayed tightly shut to cover the entire 300 millisecond window.
Wow, okay.
And the definitive proof is the bimodal condition. When trained on an environment with two peaks of danger, the mice actually produced a literal two-peaked blink trace.
Wait, seriously, a double blink?
Yes. They would begin to close their eye at 200 milliseconds, slightly relax their eyelid during the improbable middle window, and then squeeze it shut again for the 500 millisecond mark.
That is wild. The physical movement of the eyelid perfectly mirrors the statistical shape of the danger.
Exactly. They are learning the entire curve.
Okay, let's do a quick 5-minute recap for you listening to lock this in before we dive deeper into the brain tissue.
Good idea.
So, bullet 1, your brain uses Bayesian inference to handle uncertainty. Bullet 2, researchers tested mice with different probability distributions of time before an air puff.
And bullet 3, the kinematics of their predictive blinks flawlessly mirrored the statistical shape of the danger, proving the brain learns the full probability curve.
Perfect. So that's the behavior. Now let's transition to the physical squishy brain. Where exactly is this happening?
They plunged specialized, high-density probes into specific sites in the mouse cerebellum, lobules IVV, and the simplex lobule.
And the main actors in these regions are a highly specialized type of neuron called the Purkinje cell, right?
Yes, the Purkinje cell is the star of the show here. They are massive, highly branched neurons, and they are the sole output of the cerebellar cortex.
Meaning everything from that area has to funnel through them?
Exactly. Now, what's fascinating is that they exhibit two entirely distinct firing modes. It's like they speak in two different voices.
Right. The simple spikes and the complex spikes. Or SSB cakes and CSB cakes.
Let's use an analogy for that.
I love analogies. Let's say simple spikes are like a fast, continuous chatter. A high-frequency baseline hum that's just always going.
That works perfectly.
And complex spikes are like a rare, loud, booming voice. They're low frequency, but when they fire, they're so powerful they actually pause that fast chatter for about 20 milliseconds.
Exactly. So when the team analyzed that high-frequency chatter, the simple spikes, they found that as the uncertainty of the environment widened.
Going from narrow to wide.
Right. The tuning profiles of the simple spikes broadened perfectly to match, and they analyzed over 3,200 individual units using principal component analysis, or PCA.
Proving that the whole neural population physically shifts its tuning to match the statistical environment.
Precisely.
But here's where it gets really tricky from a data perspective. If you just take hundreds of trials from a mouse and average them all together, of course it looks like a smooth curve.
Because averages always look clean.
Exactly. But real life isn't an average. If a predator is jumping at you, you don't get an average of 100 jumps. It's one trial.
One moment at a time.
So how do we know what the brain is doing on a single trial basis?
This is a huge hurdle in neuroscience. Single trial neural data is incredibly noisy. So they used a deep learning AI method called LFADS.
Which stands for latent factor analysis via dynamical systems.
Let's stick with an analogy for this one too.
Okay, think of the raw neural data like a recording of a chaotic symphony orchestra playing in a room with terrible acoustics.
There's static, people coughing, instruments bleeding into each other.
Right, if you just listen to a 3-second clip, it sounds like total garbage noise.
Ooh.
But LFADS acts like an advanced AI audio engineer.
It can isolate a single violin's melody from that chaotic recording.
It infers the hidden dynamics, the latent manifold from the messy static of the brain without needing to average anything.
That's a perfect way to explain it. And the results from LFADS were conclusive.
What did it show?
It proved that the single trial neural activity metrics perfectly correlated with the single trial behavioral metrics of the mouse's blink.
So the brain is actively calculating the probability dynamically, moment by moment, on every single attempt.
Exactly. It's not just a static representation.
All right, time for our second checkpoint.
I'll take this one. Purkinje cells in the cerebellum are the main actors processing this timing. They're symbol spikes that fast chatter change their firing patterns to broadly match wide uncertainties.
An advanced AI, the LFADS system, confirms that single-trial neural activity directly correlates to single-trial blink behavior.
Spot on. So that covers the high-frequency chatter. But what about that rare, loud, booming voice? The complex spike.
Right, the missing signal.
Historically, we only knew about 2 types of complex spikes in this circuit. There was a CSP kite, which fires right after the visual stimulus.
Basically saying, hey, the light just turned on.
Right. And then the CSP care puff, which fires right after the unconditioned stimulus.
Saying, hey, we just got puffed in the eye.
Exactly. But this paper reveals a massive breakthrough finding in what the researchers called wide experts.
Meaning mice that had prolonged, extensive exposure to that highly uncertain wide probability distribution.
Right. In those wide experts, a brand new third type of complex spike emerged. They called it the CSPK prior.
And this booming voice reliably fires at the exact onset of the earliest probable interval.
It essentially marks the very beginning of the danger zone.
Now, a really important nuance here. This doesn't just happen on day one, right?
Not at all. Naive mice don't have it. Even mice abruptly switched from a predictable environment to an unpredictable one don't have it initially.
So it develops over time.
Yes, through intrinsic plasticity in the olivary cerebellar loop. The brain hates uncertainty because it's metabolically expensive, so it creates this new timestamp to mitigate that high uncertainty.
It brackets the start of the uncertain period so the nervous system can deploy its defenses without exhausting itself.
Precisely.
Okay, but again, I have to play the skeptic. We see the cells firing. We see this new prior spike. But correlation isn't causation.
It's the golden rule of science.
Right. Just because the cells fire right before the blink doesn't mean those specific cells are actually causing the predictive blink.
So how do you prove causation? You have to actively manipulate the system. And they did this using optogenetic interrogation.
Optogenetics. This is such a cool technique.
It really is. They used mice expressing a light-sensitive protein called CHR2, specifically in their Purkinje cells.
So they literally implant an optic fiber and shine a blue laser light directly onto the cerebellum.
Exactly. And that light artificially facilitates or increases the firing rate of only the Purkinje cells exactly during that critical prior window of time.
Hijacking the neurons while the mouse is doing its probability math.
You got it. And the result.
Lamp.
The optogenetic stimulation severely and reversibly wiped out the predictive blink.
Just erased it.
Erased it entirely. The prior dependent response just vanished.
But wait, could they still blink normally if they got puffed?
Yes. That's the crucial part. The reflexive blink remained totally intact. This proves the light didn't just paralyze the eyelid.
It specifically erased the brain's mathematical prediction of the future.
Exactly. So Purkinje cell activity directly drives the predictive behavior.
Okay, checkpoint 3. Prolonged exposure to uncertain timing generates a brand new brain signal, the CSPK prior.
This booming spike marks the beginning of the danger zone.
And optogenetics, using light to control neurons, Prove that Purkinje cell activity directly causes the predictive blink.
Perfect summary. Now we have the what and the where, but we need to answer the ultimate question. The how.
Right. How does a wet, squishy brain actually do the math to create a probability distribution? We aren't silicon computers.
No, we are biological networks. To explain this, the paper details a proposed computational model involving two other critical players, granule cells and climbing fibers.
Okay, let's break this down.
Granule cells act as the timers. When the warning light flashes, about 100,000 granule cells fire in a cascading, decaying temporal sequence.
They create a temporal basis set.
Exactly.
So visualize this like a massive game of telephone played down a line of 100,000 neurons.
That's a great way to think of it, but there's a catch, right?
Right. It's called Weber's law, or scalar variability. Basically, our internal clocks get fuzzier the longer the time interval.
The first few messages in the game of telephone are crisp, but by the 500 millisecond mark, the timing gets sloppy and blurry.
Okay, so the granule cells are the ticking, increasingly fuzzy timers. What about the climbing fibers?
They are the teachers. The actual air puff triggers the climbing fiber to fire.
Delivering that booming complex spike.
Exactly. And the probability math is calculated through a tug-of-war between two plasticity rules, LTD and LTP.
LTD is long-term depression.
Right.
The weakening of a synapse.
Yes. If a granule cell is firing about 50 milliseconds before the climbing fiber fires.
In a window called the eligibility trace.
Right. If it fires in that window, the synapse connecting the granule cell to the Purkinje cell gets weaker.
It's rewarding the neurons that fire just early enough to actually close the eye in time.
Exactly. And LTP is long-term potentiation. That's the opposite.
So if a granule cell fires, but there is no climbing fiber, meaning no puff happened, the synapse gets a weak restoring force, making it stronger.
It's a constant tug-of-war based on whether the threat actually arrives.
This leads to the most mind-bending analogy of the entire deep dive, to really visualize this footprint of memory.
Let's hear it.
Okay, because the air puff happens at random times in a wide distribution, LTD carves away synaptic strength across a broad area.
Right.
But you have to remember, Purkinje cells are inhibitory.
They act as the brakes for the rest of the brain's motor system.
Yes. So, because LTD weakens the synapses, it creates an inverted prior distribution in the tissue. It's like a footprint pressed into wet sand.
The LTD presses down, carving out the sand.
Exactly. And because the synapse is weaker, there is less Purkinje cell firing. Less firing means the biological brake is released.
And releasing the brake allows the blink to happen.
The physical inverted shape of that synaptic footprint is the literal manifestation of mathematical probability.
It is incredibly elegant, but impartially we have to report the paper's caveats.
Right. No system is perfect.
Both humans and animals struggle to perfectly learn highly complex priors, like distributions with multiple erratic peaks or high kurtosis.
Where there are extreme sudden outliers.
Right. Because that granule cell game of telephone acts as a low-pass filter, muddying complex signals over time.
The hardware just gets too fuzzy.
Exactly. And the authors note that other brain areas, like cortico-cerebellar loops, are likely involved too.
Okay, final checkpoint. Granule cells track the passage of time. The tug-of-war between synaptic weakening LTD and strengthening LTP physically imprints the probability curve into the brain.
And because Purkinje cells act as brakes, weakening their synapses releases the brake, causing the predictive blink.
Taking a step back, this is just incredible. We've gone from a mouse blinking at a puff of air to understanding the literal mathematical algorithms our synapses use to predict the future.
And this applies directly to you listening right now.
Every time you successfully anticipate a bouncing ball or guess that delayed train's arrival, your cerebellum is running these exact Bayesian calculations.
Weighing LTD against LTP, based entirely on your prior experiences.
It's amazing. Which leaves us with a final thought for you to mull over.
A provocative one.
If our cerebellar synapses are essentially acting as physical molds of our past experiences to predict the future, what happens when our environment changes faster than our synapses can remold?
When LTP can't keep up?
Exactly. Our modern anxieties or our constant misjudgments in this fast-paced digital world, just our brains playing out an outdated probability distribution.
That is a fascinating question.
Next time you confidently anticipate a disaster that never actually happens, just remember, that's your Purkinje cells releasing the brakes on a ghost from your past. Thanks for taking this deep dive with us.